So very much looking forward to this, and if you are one of these students and you're not completely sure about like the format, please come find us and we'll talk about it throughout the day. Okay, and so it is such a pleasure to introduce our keynote speaker for today, Nanthia Suthana. Nanthia's work focuses on the intersection of cognitive neuroscience, engineering, and clinical applications, which sounds already very exciting to me, but even more exciting is the unique research approach that Nanthia takes in her work. When she launched her lab at UCLA, she basically launched a whole new research area where she and her lab have been developing tools and pioneering work to record from deep brain regions in the human brain, such as the hippocampus, while people are freely moving around and naturally behaving in real-world everyday life situations, which has enabled such important insights into human cognition and behavior in the everyday world, which was simply not possible before. Nanthia has a long list of grants, influential papers and awards, not only for her research, but also for being a fantastic teacher and mentor. And I had the huge pleasure of experiencing this myself when I was a postdoc in Nanthia's lab for several years. And I can tell you from my personal experience that Nanthia is not only a brilliant scientist, but also a wonderful person and mentor. Nanthia, I've learned so much from you about not only the way you're doing science, but also your unique vision and approach. And I'm so happy that you're here today and can share more of this with all of us. Thank you so much for joining us, and I'm very much looking forward to your talk.
So much, Matthias. It's hard to start after that. It's really so special to be here to see, the lab be here at BU, where Matthias has started his own lab. Very proud, to say the least. And to the conference organizers, Matthias and everybody, it's just been a fantastic conference. Usually I have to give this talk to a crowd I think that is a little bit skeptical of studying naturalistic behaviors, and I really don't have to do that here. And thank you to the wonderful talks yesterday who introduced this concept. So I would usually start with saying, in order to understand human cognition, and in my case memory, we need to get out into the real world and start measuring the brain and behavior in these kinds of settings. However, this is not how traditionally neuroscience has been done, right? We have people sort of laying still because motion artifacts and various other reasons. But if we want to not only understand the brain and behavior in this context, we need to also study this in this kind of a situation in order to help treat patients. And that was really what was said yesterday. So I'll stop there. But it's been really great to see all of this work. And Matthias is very humble, but I'd like to just highlight that everything I'm showing you today has really not been possible without him and his collaboration and integration into all the projects. So each slide will have a photo of the first authors who contributed to these papers, and so there's a long list of others, including Matthias, who've helped out with all of these studies. This was an article that came out last year to really highlight the timeliness of this approach in terms of non-invasive and invasive recordings and behavioral wearables as a way to really study the human brain and behavior. And it's a really exciting time for this kind of work. So if you're interested in this, take a look at this paper. One of the things we highlight in this article is that in order to get to the real world settings, and this was mentioned yesterday, there's this big gap in between laboratory studies and real world, and we try to fill that gap in our lab as well as go into the real world by using things like virtual reality. This is an example of one of the environments that was built by our team. So our artist and our VR programmer, builds things like a grocery store, a library, a museum, to really understand human memory in a freely moving setting for several reasons. One, to fill the gap between animal studies. This is often the way that memory is studied in freely moving rodents. Several Nobel Prizes have been awarded in these kinds of settings, and so we try to mimic that scenario in humans. And we're able to do that because of the devices that have been approved and implanted individuals that you heard about in Nicole's talk yesterday. that are currently implanted in several thousand individuals. So this is an example of one of those devices and a patient who is willing to be videoed for this. So we integrate the device recordings with wearables that you saw in this video here. So motion capture sensors that are tracked by ceiling-mounted cameras, VR headsets, AR headsets, other wearables. And even though these are implanted in several thousand people across the US, that number is expected to go up in the next 5-10 years to possibly even 100,000, because what's happening is these devices are getting upgraded, the old devices are getting upgraded to the new systems which can record brain activity, such as the Percept and the NeuroPace RNS. So in these participants who come to the lab, in many cases they wear a lot of different kinds of wearables depending on the purpose or the hypothesis of the study. So we use biometrics such as heart rate recordings, skin conductance, respiration belts, eye tracking, pupillometry. And then for simulating the environments in our virtual and augmented reality studies, we use these headsets. And then, every year there are several new things coming out, and we explore, the benefits of those as well. So how do you get the intracranial data synchronized with the external? This has been the challenge since I started my lab in about 10 years now. Every day, this is the problem that you all experience and we experience is synchronization. This is a very challenging problem, and the way that we solve this is by creating a bunch of accessories, that allow us to integrate all these signals, such as Raspberry Pis, off-the-shelf components, and put them all in a sort of backpack with cables that we try to minimize. It looks like this. Now, the very first version is a slightly clunkier version, but the full version that you see with this wand that allows us to wirelessly view and record the data can allow for millisecond synchronization. And that's really what you want to get when you're looking at these intracranial EEG signals or LFP signals with high temporal resolution. There are other forms, such as a lighter version, which we've used to enable less accurate, maybe half a second synchronization, but it's really something that's wearable and that could be used at the home setting. And just to let you know that we're working, I'll show you at the end of the talk, on a flex coil cap that turns all of this into a cap through the Brain Behavior Quantification and Synchronization program at the NIH, and so we're very excited about that. Okay, so this just shows you an example of what an experiment looks like. It's actually Matthias's study that you see here when he was a postdoc, and Matthias is probably either standing on the outside or somewhere in the room, and the participant is walking around looking for these locations. It's a memory task. And Urush, one of our engineering students at the time, helped to basically build this backpack and allow us to record the data that you see on the bottom right, 4 channels in the hippocampus, very clean data, free of motion artifacts, because this is a fully implanted shielded system. On the top right, you see their gaze position, their eye movements through one of these eye trackers, and on the bottom left, you see their position in the room from the motion capture cameras. On the left, you're seeing an example of one of these depth electrodes. It's right in the hippocampus. And so because of the placement of these electrodes, we're very interested in studying episodic memory or spatial memory in this context. And I told you that this is most often done in freely moving rodents. So for example, place cells that you've heard about that won the Nobel Prize, grid cells, they're tightly linked to these oscillations that you see on the bottom, a particular frequency called theta, theta oscillations around 5 to 9 or ranging around that area. These oscillations are very important to memory. If you block them, an animal can't really function through a maze as well. Their memory is impaired. And no one's ever recorded these oscillations, or place cells or grid cells, in freely moving humans. So that was our first goal, is to really try to see if they exist. And you see some of the data here, and if you're familiar with theta oscillations, you might say, oh wow, okay, there are some theta oscillations in there. But we wanted to quantify this. And Matthias came along and had a really brilliant idea to do this in participants who are engaged in a memory task. So on the left, what you're going to see is a participant walking. As they get closer to the wall, the theta power, which we're quantifying using these BOSC methods, better oscillation detection methods, going to go up as the participant gets closer to the wall. And so you see them approaching the wall, and the signal in theta band goes up. It's very specific to theta. You also see it in gamma. You also see some cross-frequency coupling, and it goes down as they go away from the wall. Now, the really smart thing about this task, when Matthias came to me and had this idea, and I thought, let's run this tomorrow if possible, is that he decided that it would be interesting to have the participants sit in the corner of the room on the right, you see, and have them watch him do the task and walk around. One thing I failed to mention is that this task is a spatial memory task. They have to learn different locations. And when you do this in a small room with these walls, the walls end up being very critical for how you accomplish this task. And this is one reason why we think the walls, the proximity to the walls are serving, as a very strong modulator of this theta signal. So Matthias is walking around. You can see the participant's eye gaze position on Matthias as he's getting closer to the wall. And what you see is that theta also goes up as Matthias is getting closer to the wall and goes down as he gets farther away. This was a very consistent signal across all the medial temporal lobe channels and participants that ran this study. And there's a lot of more interesting stuff in here. It's probably the longest supplemental materials I've seen in a paper that you can explore that's been published a few years ago. So this just shows the quantification of the theta signal increasing as they're close to the boundary walls and smaller as they're in the inner room area. And interestingly, on the right, you see as they're sitting, so they're not moving and watching Matthias get closer to the wall, you see the same effect. So this theta activity can be modulated by the self position as well as another person's position. And Matthias, as well as several others, including Zara Agajan, who is now at Kernel, who was my first postdoc in the lab, characterized these theta bouts. And what's interesting about this activity is that in humans, it comes in bouts. In animals, it does not. And this was a big source of sort of controversy when we first published this results, because in rats, it's really continuous as an animal is running. But in humans, you see them coming in bouts, and it's a very small prevalence, about 10% of the signal, but it's modulated by behavior. So it's modulated by proximity to the wall. In this case, it's modulated by walking speed. So the faster they're walking, the stronger sort of the prevalence of these oscillations is. So on the bottom right, you can see as movement speed is going up, that 8 hertz oscillation sort of happens more often. And in several other papers, we've replicated this finding. And so I think the community, at least in the animal research community, is starting to believe these findings, especially now that other species, like non-rodent species like bats and non-human primates, are showing similar effects. Okay, so this task that Zara ran did not have a memory component, and so what's interesting here is that when there's no memory component, speed seems to modulate these oscillations, but when Matthias did a study that had a strong memory component, speed was no longer the contributing to the variance of that signal, rather proximity to walls. Now Martin came along and did an experiment where the walls are really, really far away from the task, and so they're maybe perhaps not as relevant. What's relevant in this task is when and where to turn. And so we had a spatial memory task where participants had to learn a walking trajectory and very distinct turns in the trajectory, either to the right or the left of this center track, which includes a treadmill. And I'm going to show this to do a couple things, which is one, convince you of the infrequent bout nature of these theta oscillations, and also show you how clean and clear these signals are that we can record. And so what you're going to see is that theta, you can see by eye, the bouts increase in amplitude right before the participants make these turns. And again, these turns are critical retrieval points for this task. There's a theta bout, and there the participant is turning. Another one here now, not even close to walls. There's a theta bout, and then the participant's turning. And I'll show a few of these trials. Do I have a pointer? Yeah. All right, so there, right here is a very strong theta bout, and then right following that, there's a turn. Okay, so this was the performance of these participants. They look on a iPad basically, of a walking trajectory, and they try to replicate this trajectory trial after trial. They get feedback. And you can see these participants are doing very well in this task. They're learning these trajectories. There's 4 turns in the rightward trajectory and the leftward trajectory. There's one participant, if you can notice, P2, that for whatever reason, they added this little extra turn, which at the time I'm sure was very frustrating for Martin because he wants everybody to do the task the same way. But it ended up being very interesting. So on the next slide, I want you to sort of pay attention to that little extra turn. And what you're going to see on the next slide is the heat map, basically, of theta activity on these trajectories. So you can see in the group level, you've got increased theta power right before these turns, and it's very striking. And every single participant shows evidence of this, even in participant 2 with that extra turn, which ended up being a lucky sort of happenstance. You see this increased theta power. I can show you individual trials. You see it very clearly on individual trials that these turns are modulating theta. Now, the third trajectory, which I probably won't have time to talk about, is not as strongly modulating theta by these turns. And it turns out there was a dual task on that third trajectory. If you're interested, I can talk more about that. Now, what's interesting about this study is that Martin decided to have a treadmill in the middle of the room. And the purpose was was to have them walk on the treadmill to control for speed. But instead of having them walk through the room in these turns, imagine navigating those turns. So he would cue them with, okay, I want you to imagine the rightward trajectory, leftward trajectory, so on. And normally when you imagine experiences, you speed up the process in your mind, so you do it a little bit faster, you compress. And he really enforced them to take the full time. So if they went too fast, you would say, okay, slow down. Try to imagine the walking trajectory as if you were really experiencing it. And what he found is that there was some evidence for this increased theta power happening in these imagination trials where the temporal consistency across trials was significantly above chance. Now, how do you determine this? What's your control condition? And this was a challenging thing to do, but what he did is he had people walk the treadmill before introducing the concept of imagination without imagining. So just walk on this treadmill. And he controlled the speed, and so they're basically matching for their speed, but there are no external cues that are changing, right? So the control condition is just walking on the treadmill without imagination. But during the Imagine trials, you start to see increased correlation across trials, and he explored, what is driving that correlation, and it turns out there is some, basically increases in these theta bouts, not perfectly aligned to, the trajectory, but some evidence for that. And if you look at the correlation during Imagine navigation by channel, so each individual point is individual brain channel, and you look at the real-world navigation, you see a correlation such that the channels that show the highest temporal consistency across trials during real-world navigation also show this during Imagine navigation, and those electrodes happen to be in the anterior hippocampus, one in each participant. The other electrodes are in areas like the entorhinal cortex and others. Okay. So this study suggested that these theta bouts can be modulated by critical retrieval points, and that they can be also reinstantiated during imagination without external cues. Sabrina, MD PhD student, came along and wanted to use VR because VR has the power to easily change the context, and wanted to know if I switch the context, for instance, going from a concrete room to a wooden room, but the layout is matched, but the cues are different, will these, theta bouts, these patterns of activity, remap to the new context? And so she designed an experiment that allows people to learn various locations. You see these colored cylinders in the room that they had to remember, and then recall those positions by using a handheld controller where they press a button every time they are near one of those remembered locations. So what you're going to see here is their trajectory followed by a cross, X, for their recalled location, and then they get feedback. So on that trial, they did very well. They revisited the position where the halo was, in fact. Then they get some control trials, which are these arrow trials, where they see arrows, and they're just cue-driven trials. And then they go again and do the next halo, and on this trial, they were a little bit farther away, so they didn't do as good of a job. So she was able to characterize when they were correct versus when they were incorrect, depending on how far away they were from the actual location, and she found that these 6 to 8 theta hertz oscillations increased in power whenever the individuals were actually, in fact, closer to the remembered location, basically more accurate. So this theta activity was modulated by correct recall. It was not significantly occurring during the incorrect trials, and it was also not occurring during the visible feedback time periods or the control arrow trials. To look at the time course of this increase was happening right before the dotted line here, which is when they were pressing the button on the handheld controller. If you put all these variables in a linear mixed model, and not all of them are shown on the right, you see that performance really contributes the most to the variance of this signal, such that if they were correct or incorrect, is significantly higher. Okay, so I made the choice today of just highlighting a little bit from every study, but these are all well, most of them are published. Actually, Martin's is also pre-printed, so you can explore, more. But I want to also highlight Corey Inman, who is a postdoc in the lab and now is assistant professor at University of Utah. He decided to take this platform outdoors, which is very challenging. He had participants walk South Campus of UCLA, which is the very confusing area if you've ever been there. I've worked there for 20 years and I still get lost. And he had them learn a trajectory. So they had to do this several times and learn a specific way, doors to go into, turns, etc. So he has, 100 hours of recordings of participants walking around and learning these trajectories. So sometimes they're outdoors, sometimes they're indoors, sometimes they're going through doorways. We're doing eye tracking so we can see what they're looking at. Sometimes they may talk or engage or get lost all of a sudden and do this sort of behavior, which ends up being very interesting for analysis, where they're trying to figure out where to go. So I'm just going to show you one result from this one, which is that overwhelmingly in all the channels, the signal in the low frequency range, especially in this theta range, was higher when the individuals were walking outside versus when they were indoors. And we're now exploring why this may be the case, and there could be very, very different reasons for this. So we're looking at the video recordings, we're looking at certain landmarks, event boundaries, etc. I do want to show you one result which seems to be consistent with one of our previous studies, such that when they're about to turn, you see increase in these low frequency oscillations and these dips after they contribute. After they make that turn, that's sort of independent of their speed. They're actually slowing down here. Okay, so to summarize this first half of the talk, because I want to go into the clinical applications, we see that these theta oscillations in humans, they come in bouts. They're infrequent, about 10% of the signal, which is very different than what we have seen in animals, in rats, excuse me. But it happens to be consistent with bats and non-human primates. Zara Agajan showed that these signals are modulated by walking speed. So this task was just simply walking back and forth in the room and in circles. And when there's no memory component, it's modulated by speed, such that the faster you walk, the more prevalent these signals are. In the 8 hertz frequency band, also in 16 hertz, which we think is related to a harmonic. Sabrina came along and found that this theta activity is modulated by successful memory. So not just memory, but when you are actually in fact correct. Versus not correct. Matthias came along to show that these signals can be modulated by other variables such as proximity to the walls when you have a memory task, and can be modulated by the position of another person. And this had never been seen before, even in animal studies, in terms of this theta activity being modulated by another conspecific. Hamza Zubair, who's a MD PhD student right now, is exploring eye movements, and what he sees is that these theta oscillations are aligned to saccades, and this makes sense in terms of how we navigate the world versus rats who are using whiskers. But the interesting part about his findings is that they're aligned to saccades, but only when there is a strong memory component in the task. Martin came along and found that these bouts of theta can also be there's evidence that they can be sort of recapitulated during imagination and not without external cues. And then Corey is exploring these activities during outdoor navigation and spatial memory. Okay, so with that, here's another summary. I pretty much said all of this, I believe, so I will go on to the next slide. Okay, so as we were developing these technologies to study memory, which has been my fascination since I was 19, and discovered sort of, the concept that there's this brain region that can completely, when damaged, can completely obliterate your ability to form new memories for your personal events. That was what sort of started me along this journey. I realized that these tools could be useful for other things. So we provided these tools to other labs, and I want to highlight 2 of those for you today. So the first one I'll highlight is Katie Cross at UCLA, a neurologist who works with Parkinson's disease patients and how we're using that. And then the second is PTSD. I also want to mention that we're recording studies during sleep, during naps in the lab, which is very important for memory consolidation. And we're also looking at observational learning and 2-person learning. So if there are 3,000 individuals currently with these devices, you can imagine that we could bring in 2 people at a time. And so I'm very excited for the possibility of looking at 2-person interactions and have had many discussions, some discussions with Guillaume, who you're going to hear about later today, to look at, potentially how these oscillations may be synchronized in individuals. Okay, so Parkinson's disease, Katie Cross is working with patients who have electrodes in the subthalamic nucleus as well as other areas. And so we designed a VR task here to elicit freezing. Freezing in Parkinson's is very, very challenging in the clinic. At home, patients will describe having episodes of freezing, and then when they come to the clinic, we'll have some challenges in, replicating this because the normal, everyday, real-world triggers are not present. So what we did here is we have a real-world task that we try to elicit freezing, such as going through narrow doorways. You see a barn door that was installed in our lab where we can manipulate the size opening. We have an alarm system that goes off that creates some anxiety for the patients. We have an elevator with people coming out, not a real elevator, but a virtual elevator with people coming out. We put furniture in the room. We do all sorts of things to see what, on an individual patient basis, can trigger their freezing. And so here you see one of the conditions where the doorway is made smaller and the patient will freeze as they approach this doorway. We're recording from the subthalamic nucleus as well as the scalp EEG headset, and you see one channel of each on the bottom. And so the reason we're doing this is to try to develop a closed-loop simulation therapy to prevent freezing. And so what the signal that we're looking for that can predict freezing and then trigger the stimulator to turn on and hopefully ameliorate the symptoms. So this is a study that's ongoing, and so I'm not going to show you too much else, but very fascinating work that Katie's doing. The next one is PTSD. Now this, from a personal standpoint, was interesting to me because of the memory component, right? So these are patients with memories that they cannot subdue or inhibit because of the traumatic nature of them. And so what we're doing in trying to understand the neural correlates of PTSD is designing, again, to bridge that gap between animal studies, designing a task that is often done in animals to study PTSD, and that's fear conditioning. So I won't tell you much more than that except to show you this video. So participants are walking through an environment. There's a color change that might occur. And they're asked to, basically just, so there's something that happens here. It's much louder when you're wearing the headset. I wasn't sure if the sound was gonna work. But when you're wearing the headset, what happens is there's a loud sound. Obviously, a spider jumps on your face. And the headset actually vibrates a little bit. So what you see is people get pretty startled during this experience. Here's a blue light. Nothing's going to happen here, so don't worry. It's a safe cue. We don't tell them this association. They learn this over time, that the blue light is going to predict safety or not, a neutral scenario, and the green light predicts these aversive stimuli. So this is a way to test fear conditioning in the lab. We measure skin conductance, heart rate, and other biometrics, and again, here something's going to happen, just to warn you. And we do this several times. Actually, we've let go of the bee because the spider ended up being more aversive. But what we found is that can I pause this? I don't want to keep showing you this. Okay. So, so Michaela, who's an MD student, is in the video, and you see her just like sort of walking and not getting startled at all. That's not normally the case. She's done this many times. But when participants come naive, they do end up getting startled, some more than others. And it turns out that individuals who have a very high sort of trait anxiety score on a questionnaire that you can give them afterwards show a really much higher response in their skin conductance. I'm probably over here because I have a lot of anxiety. And so you see a correlation with their trait anxiety and their skin conductance response. Now, what's happening in the brain? So if we bring in patients who have amygdala electrodes, so now this is the amygdala, not the hippocampus, what you see is that theta power, a slightly reduced frequency, actually around 6 hertz, 5 to 9, increases when that spider comes up. So right after that spider, about a half a second, you see an increase across all the channels. You're seeing individual channels to the spider. And that over the course of this experiment, as they start to learn these associations between the green light, which we're calling the conditioned stimulus, and the blue light, you see that response starts to happen right after the green light, so before the spider, right? And so it's predictive of the unconditioned stimulus, or US, which you see right here, compared to the blue light. And we counterbalance the light and so on. Okay, so this is a way to bridge the gap between the animal studies, try to understand the amygdala's role in this process, but does it have any real life, implications in terms of treatment. And this is where we decided to go with PTSD. So there are some patients with PTSD who are refractory, they're treatment resistant. They do not respond to medications, exposure therapy, cognitive behavioral therapy, et cetera. And these are the patients that we were looking for. So we work with the VA, the Los Angeles VA, and the clinical trials for 6 combat veterans. We've implanted so far 4, we have consented 6, and so probably by the end of this year we will have completed this trial. The goal of this trial was to try to detect the signal that predicts these aversive events, such as a spider, but in the real world, so when they're triggered. When they see fireworks, for instance, a lot of these patients have fireworks as a trigger. When the device records a signal, have it stimulate to prevent the behavioral symptoms. Okay, the way we, so what I want to show you first is our first patient, a video describing, and maybe if the volume can go up a little bit, or if you can hear, hopefully you're going to have him describe his experience before the trial. So this is a common experience for these very treatment-resistant PTSD. These are all combat veterans that we included in this.
And all of a sudden it just hit hard with a ton of bricks, where I started seeing things that weren't there. I thought it was just all normal stuff coming back to me, like visuals and memories and everything, but it got real intense. Be on like a patrol all of a sudden, and I'm not on a patrol, I'm in Target. I've had hospital visits, I've had times where I've been blacked out on the ground, and ambulance care guys are just sitting there like trying to figure out what's wrong with me. They don't know what's wrong with me. I couldn't express what it was going on. I just knew it was like about war.
So this is a common experience, like I said, in all of our patients, these very traumatic experiences that they describe that were related to some incident while they were in combat. And so what the approach here was that we would have them come in and we do a series of tasks. One, for instance, like looking at emotional war-related images versus positive images versus neutral images. This one was a very difficult one to do. We would actually ask them about their trauma, so they would describe it, and we would create a script of their trauma and present it to them. We'd also present a pleasant memory as a comparison. And then the third one is for them to actually mark events at home. So the device we're using is the RNS. It has one very interesting capability, which is that a patient could swipe a magnet over the hardware that's underlying and implanted, which will trigger storage of the data. And the way the data is stored is it goes back in time. So you get a few minutes before the magnet swipe. And so you know, if a patient gets triggered, let's say there's something on the news, the study was done during the time when the Ukraine war was happening, so that was a trigger many times. The patient could take the magnet and just swipe it over and save that piece of data, and then we can look at it. It goes to the cloud, and we look at the data. And so these were at-home symptoms. We also had scheduled recordings as controls to happen during periods when the patient reported, you know, sort of neutral periods. Okay, so across all of these 3 tasks, the red is showing you the aversive periods. So the negative images, the traumatic script that is being replayed, and the at-home triggers, all in red. And in all of those, there's not a lot of data because these are 2 patients, 4 channels in the amygdala. Activity is increasing, you know, in this theta power, but again, this is the amygdala. So it shows you on the left an example of a channel in the amygdala. On the bottom, you see a heat map of basically that theta activity that's happening after the onset of these images. We had a cohort of several epilepsy patients who also had amygdala recordings, but you know, did not have PTSD. And for this, we showed them these IAPS images, and we saw no differentiation. And this is actually a replication of a previous work by Jacqueline's lab. No differentiation between the negative and the positive neutral images. Suggesting that maybe in PTSD, and this is again preliminary, that there is this increased sort of or lowered threshold for this amygdala theta power activity for negative or aversive you know, experiences. Okay, so this was very exciting to us because it was concordance across lab-based tasks, more naturalistic, and then really real-world experiences at home. And so the strategy was then to program the stimulator to turn on every time it saw increased power in this frequency band. And this is just to show you that it really was specific to theta. So looking at all the other bands, you don't see this effect. You see it only for theta activity and only in these PTSD patients, which is shown in pink, and not in the non-PTSD patients. And on the right, you're seeing the PTSD patients versus the non, and you will see this very specific bump there. All right, so the stimulator was turned on. after 3 months, 3 months after implantation, and what happened. So this is the first 2 patients. In the purple, you see their CAP score. So this is the clinician-administered PTSD scale score. Those of you who are psychiatrists are very familiar with the score, and a high score is worse. And the first patient, actually both patients, were starting in the 60s, right? So very, very high CAP score. And right around when the stimulator was turned on, so what you see here is month 3, right here, actually month 2, around there, the stimulators turn on, and the first patient, you see a really strong, not immediate as in like hours or days, but the scale on the x-axis is months. You see a drop in the clinical score. This is a clinician-administered score. There's also the PCL, which is the participant-administered score, which also decrease. And there are various subcomponents of this score that you can look at, such as re-experiencing was one domain, which was being reduced quite significantly. The dotted red line is the remission level. And so this patient very quickly ended up going down into remission and has stayed there for now almost 3 years. The second patient had a little bit slower of a trajectory, and there are some interesting differences between these 2 patients. Just to give you a sense of the stimulation, this is a very traditional stimulation that's used for Parkinson's disease, but again, it's in the amygdala. The amount of stimulation is very small, so it's 90 minutes per day spread out, really when this theta, low frequency activity is increasing in the amygdala. So the patient on the right had a slower trajectory, but you can also look at the blue graph, which is the number of stimulations per day, which was also slow to rise in the beginning. And it turns out that that patient's electrode stimulation was programmed slightly differently, meaning that it only turned on when both hemispheres showed this signal simultaneously, whereas the first patient turned on when either or. And the reason they did that from a clinical standpoint, they had some concerns about overstimulating the second patient. Once they found that this was safe and that they could proceed with higher levels of stimulation, they reprogrammed that patient to be more like the first, and so now they were getting a lot more stimulation, and as that stimulation was going up, again, still very little amounts of stimulation. As that stimulation was going up, the patient scores started to go down, and this patient is very close to remission as well. All right, now I can show you numbers and I can look at numbers all day. In fact, that's what me and my lab, we do, but it's really not the same as talking to the patient. Like, how has this been for you? What's it like? And this is him describing his experience after the trial.
All in all, the experience has been wonderful. I came in with little hope because it's just the way that things were going in my life, and we put the device in, it got turned on, and then like a couple days later, I started getting these funny little like tingling sensations throughout the body. It's the washing over feeling, like you've been run over by water. I had been triggered pretty severely actually at that time, and it all of a sudden just like cloaked over me, and I was fine.
This was really impactful for me to hear this experience, because like I said, it's really, you don't get this from the data, and seeing his progress and transformation has really sort of changed, how I think about the value of science and, translational work. So we're very happy that he was able to do, basically much better. He hasn't had these episodes, these dissociative episodes. In fact, one experience that he describes here was actually taking his kids to Disneyland and watching the fireworks for the first time. And when the fireworks first started, he did feel very anxious and ran into a store, worried that it was going to cause a dissociation. But as he described, basically was able to go out and finish watching the fireworks. So it's been quite the experience, I have to say, a lifetime, once in a lifetime experience to see this. And I'm just hoping that we can replicate this for more patients. Okay, so this is one example of how these tools can be useful for not just understanding naturalistic behavior, but really treating these patients because of the capability of these devices to stimulate in a closed loop manner. Okay, last but not least, I just want to tell you about some ongoing directions that we're exploring. These LFP signals are very useful. You can record them for years, and they're very clean. There's no motion artifacts because they're implanted, but we cannot record single unit activity. And so in order to fill the gap between animal studies and also potentially get even more fidelity and personalization on these patients, there may be a period where single unit activity could be very useful. And so about 12 years ago or so, I started working with Dan Markovich and Itzhak Fried at UCLA to develop a system that could eventually be implanted to record single units. This is called the Neuroscience system. The current version is handheld that you see, which we've used to record patients walking through the hospital hallways. These are the acute epilepsy monitoring patients. You can see 12 channels of recording, or you see spikes and single unit waveforms in the bottom right as they're walking through their hospital room. We use an eye tracker and motion capture to look at that data as well. And so this was very exciting to us to record these units with, obviously some challenges with motion capture because there are external wires, but with the ultimate goal of implanting this permanently in individuals who, such as epilepsy patients and so on. So the exciting part of this device is that all the integrated circuit chips are implant scale. So they can all fit in a single pill-sized device. In fact, that's the actual device that can record 256 channels. It can record single unit activity. It can do phase lock stimulation. It's basically a scientist and dream in terms of the ability to use these devices. The trajectory for FDA and all that is very long, as many of you also know, so we're not there yet, but we have recorded signals from the externalized version, the Neuroscience, in participants who are walking, and we recorded last month an individual who's doing a spatial memory augmented reality task in the hallway of the hospital. Okay, so our goal is to get single units and local field potentials along with all these behavioral measurements in environments that are more naturalistic, such as using virtual reality experiments in the hospital ward, but eventually getting a fully implant scale such that we can record these signals in humans every day, 24 hours a day, while it is also treating their neuropsychiatric disease. So it's a win-win both for the patient and for the scientists if we can get here. And so in order to do this, it's an exciting time. There are devices coming to market, as Roy and Nicole highlighted. Every sort of year is a new company or something that's coming out in terms of the wearables and the implant scales. Thanks to Nicole for this. Basically, I adapted her slide here that you might have saw yesterday. And then one last study that I'll highlight is. Something that we're doing at home that we just got IRB approval is to use these Meta Ray-Ban glasses, which can record 3 minutes of video audio recordings with these patients who have implants, such that they could in fact record an experience like this, or a social interaction, or a birthday party, or so on and so forth. Now, this sounds, maybe it doesn't sound easy to do, but it's not easy to do because the implant, is not accessible by you can't connect to it and insert digital signals to synchronize. So how do you synchronize these glasses with the implant? And so this was a technical challenge, but I think we've solved this challenge, and I'll show you one slide on how we can do that. So the protocol for this study is to record on the glasses. So there's a little button, there's a little light that turns on that they press. And then after, a few minutes, not swipe the magnet, but they're wearing a cap with the magnet on it, which will insert a synchronization signal and also trigger the storage. And remember that storage goes back in time, so you get several minutes before the onset of this magnet and a little bit after. And you have an alignment signal, so you have this artifact that goes in the signal, in the data. We're looking at this for autobiographical memory. In neuroscience, we do not capture brain activity while people are forming autobiographical memories. We put them in a scanner and we ask about their memories, right? But we have not been able to capture these memories in the naturalistic world. And so this technology can do that, right? So it can actually go out there and capture these moments as they're being formed, and my slides are not advancing anymore for some reason. But there's only one more, so if you can manually advance them for me. So what we're doing is we're capturing these autobiographical memories, and then we put it on their phone and use an app called Hippocamera, which Morgan Berens has developed to replay these memories to them while we're also recording. So recording encoding and retrieval of memories, looking at very interesting things there, and also bringing them into the lab and asking about these memories over several months. And they capture many, many memories over the course of these several months. So we have some sort of technical problem. But the last slide was, maybe it's good that I don't show it because Nick, who drew it, I didn't tell him I was going to show this, but it shows you the technical challenge in getting this to work, which is just basically a hand drawing of Nick's drawings of how to get to solve this problem of alignment and synchronization between the glasses and the implant. And so with that, I did want to show the picture of all the wonderful people in my lab, which is going to come up. And that's really the most important slide. My lab is a very interdisciplinary lab. I make sure to hire very different individuals who are from backgrounds in engineering, neuroscience, clinical, medical school students. It's the one slide before this. This is all the academic collaborators. I work with neurosurgeons, neurologists, psychiatrists. These are all the wonderful people who do all of this work. I have an artist, a VR programmer, and this is the magic that makes all of this happen. And thank you so much to Matthias, who was very critical to this entire operation, and Zara, my first postdoc. So with that, thank you all. And just one announcement on the last slide. If you go to 2 slides, I have my email if you have any questions. And just to mention that email will no longer work next year because I am moving my lab to Duke. And so we're going to start this work there, and very excited to collaborate. We do all of this work not just for us, but we hope that others will adopt it. The problem is it's very challenging. Can you go actually 3 slides back? Because I want to show you how challenging it is. This picture really shows an example of the challenges. And so I've now realized why it's so difficult for other labs to start this work. And so my next 5 years, hopefully, is to just really, go to basically the hand drawing that looks really messy, which you're not going to be able to see anything on it. But this is just the first drawing of getting this to work, synchronizing between an oscillating electromagnet that fits in a cap. So you have to do a flex coil, basically a 2 millimeter silicon cap to get this to work. So it's very, very challenging work. But once you do it, it's done. And then everybody hopefully can use it. And so that's really my goal for the next 5-10 years. There are these patients in every major hospital, in every major city, thousands of them, and nobody is working with them. And so that's also a point that's sort of very sad to me, that more people hopefully will take advantage of this research opportunity and clinical translational opportunity to test out and develop new therapies. So with that, thank you. We can end the slides. Thanks.
All right, thank you so much for this amazing talk. It brings back a lot of great memories.
And challenging times.
Yeah, I'm sure there are tons of questions, so who would like to start?
Amazing work. I really wonder, did you also look on the eye power gamma and the relationship to theta? Can you say something about it?
Yeah, he could probably say way more than me, but basically that's what he found, is that there is coupling between gamma and theta, such that it's cross-frequency coupling, such that the amplitude of gamma is going up at particular phases of theta. And this is a finding that's been found in animal studies, memory studies, such that it's higher when they're performing better and so on. So we do see that. We see the gamma is modulated by things like proximity to boundaries. It's not as consistent as theta. And it is co-modulated by theta. I just didn't have time to go into it. So yeah, great question.
Okay. And another question, giving all the multiple dimensions, walking speed, turning, close to the room, do you have any speculation about what is the functional role? Is it like reward signal or something more general than all this?
Yeah, I mean, in all of these, again, I didn't show you, we do put it all into a model to see what is sort of winning in the sense of explaining, and it depends on the study. So in Matthias' study, for instance, proximity to walls was very, very strongly modulating the signal, not speed or other things. Eye movements, however, was also contributing. And it really depends on the task. So the difference in all of these studies is really the goal of the task, and whether that behavioral or environmental variable is really critical for accomplishing that goal. So I think it has something to do with, task relevance.
Yeah, that was my feeling, and because I saw the context effect,
Yeah.
It depends on the context and the task.
Right.
I was thinking maybe it's like the reward of doing the task right, or something more general.
So in the period where they get feedback, so we don't see, it's not happening right there. But that is a good question. I think we have a task right now that's looking specifically at reward and prediction error and also changes in the value of the reward, and I think in that setting we'll be able to tackle that a little bit better. But here, I'm sort of leaning towards this idea that this signal is very much related to basically successful memory in the context of the task. So yeah, when the context changes, it remaps to the new context. So it's it's context specific, and it's modulated by these moments, these critical retrieval moments when they're successful. So that would be a more general way of thinking. But I don't know. This is sort of new territory, because these signals, although you see them while people are sitting down, which Matthias saw it, you don't see them to the same extent, especially at this frequency range, when they're stationary in the EMU, for instance, and doing like memory task on a computer screen or in a VR task. So there's something about this experience, whether when they're moving and they're engaged, that seems to modulate these higher frequency theta oscillations. So I guess I'll stop there. There's a lot more I could say, but it's speculation. Yeah.
Thanks for the amazing talk. I'm super excited to see all this happening. And I was also curious about high gamma activity. Do you know if it's actually coming from entorhinal cortex, or is it propagated further to other structures? Because the only non-invasive medial temporal papers, one of the only ones that look at entorhinal cortex activity, is also observing high frequency gamma modulation in an eye tracking study. So.
It's a great question. I don't know that we can answer that fully yet. These first studies were, smaller sample, mixed entorhinal hippocampal studies where we do see these effects. Some effects we don't see in entorhinal, but as far as I know, the gamma hippocampal effects were both in the entorhinal cortex and the hippocampus. I think we need more studies to look at regional differences, and that's one of the goals of the next, sort of year. And to explore, even cortical. We have scalp EEG signals where we can explore theta and gamma, and there's a lot of things we need to do. So it's a great question. I think we need more work on that.
Hi, thanks for the really interesting talk. I think the work is very rich, and there's a lot of clinical applications. I had a question more on the cognitive process side of things. I think some of the tasks that you were showing, like spatial navigation or representing others' mental states. They're pretty complex cognitive processes, and I think the field is kind of interested in those in the context of it's not just the hippocampus that subserves that process. It's part of a larger cortical network. So do you guys have any data on the broader networks to which you're recording from, or do you have any interest in looking at kind of more systems level activity?
Yeah, I mean, this is why we have high density. We have 64 channel scalp EEG that's put on these participants during these studies. We've started to look at this data. We definitely see some frontal hippocampal sort of interactions that we need to explore further, but absolutely, I'm interested in this. We just as a sort of lower hanging fruit, because these electrodes are often in the hippocampus for reasons that are clinical, right? They implant these patients. There are a smaller group of patients with electrodes outside of, anterior cingulate, parietal, frontal. And so, if we kind of look at larger samples, we could explore that. But within a single patient, the only way to really do this is with FNIRS or scalp EEG with the implant. And I think that's a very interesting direction to go. Yeah.
Thanks.
Thank you again for the really fascinating talk. I just had a question about the theta bouts. What do you believe is causing the difference between what we see in mice versus in humans?
Great question. Thank you for asking that. No, I think, if you think about how rats are navigating, right, they're using their whiskers and they're sampling the environment pretty continuously. It's very different than how we do it, right? We're walking around, we're making saccades, we're doing, sort of visual exploration. Sometimes we're not even paying attention to what we're doing, and we're thinking about what we had for breakfast this morning, right? So, and these overtrained rat studies, are very different than sort of how humans can accomplish this task. So there are lots of reasons sort of related to environmental sampling that I think could be relevant here, which is why we're looking at saccades, and perhaps why non-human primates, for instance, they don't have these same continuous theta activity sequences. So I think that may have something to do with it, but we'll have to see.
Yeah, and just one more kind of related question to that is, did you look at the duration of the theta bouts, and was it significantly variant between trials, or like accuracy of memory?
Great question. Thank you for that. They're about 2 to 3 cycles, so they're very short, and they don't seem to be modulated too much. They happen, instead, they end up being more prevalent, so they happen more often rather than lengthen or shorten.
I see. Thank you so much.
Sure.
We have a question from online. Are you able to look at sleep in PTSD patients, and can you see their nightmares in their neural signal? And the second question is there potential to modulate these events without them needing to wake up?
That's a great question. Okay, so the first one. So this device, there are limitations, obviously, to the device. This is why we want to get the last device I showed you. So it cannot record continuously during sleep. It will only record if it's triggered to store the data, which could presumably happen during a nightmare, right? Or we schedule it. So we want every day at 1 a.m., but that's not going to help with nightmares because who knows when they're happening. So I don't have an answer to this question. I would like to, however, do a little bit better with the sleep characterization at night, and also have these patients, when they wake up from these nightmares, basically swipe. That would be one way to do it. This population is it's very challenging to get them to do those swipes, because when they are triggered, it's just a very difficult time. So we miss a lot of those moments. And now there's something else that's going on, which is a good thing. They're not having a lot of nightmares after the stimulations turn on. In fact, one patient reported as this was one of the biggest benefits, is that they're not waking up with these nightmares. So we can't record, it would be difficult to do this study. I've thought about it quite a bit. But during the beginning periods, yes, those first few months, every time they have a nightmare, wake up and swipe and report the details or the contents of the nightmare. And if you get enough of that data, it'll be very interesting. There's also a cohort of epilepsy patients who have PTSD. Mm-hmm. Imagine these numbers, right? If you can get access to them and have them do sleep journals and swipes at night. So I think there's a way to answer this question. It's just we haven't had the bandwidth, and so if anybody's interested, yeah, it's a very, very good one.
Thank you.
Great question. There was another one too that you asked. Did I answer that?
It's about the modulation. So would you be able to modulate these events? But I think...
Yeah, I guess that's sort of inherent in the answer, is that would be the goal, is that it's basically, if the closed loop stimulation is turning on and the device is turning on during these nightmares and then stimulating to try to prevent it. One of my goals by bringing the patients into the lab is to understand that process in a more controlled setting. Like we have a nap room, get them to sleep, or do these fear conditioning studies to try to bridge that gap. I think that question might be better answered in the lab, where you can tightly control the stimulation in the lab while you're recording. So anyway, sorry, I can go on about this, but I'll stop.
Thank you.
Okay.
Thank you. This is a great talk, such a wonderful bunch of data. Just one question about the data that you're looking at, and then a question about the next steps. I wonder if you look at microsaccades in the data?
The resolution of these eye tracking devices are not. We'd probably have to do that for the mobile. Mm-hmm. I don't think we have that, right? No.
And do you anticipate that more of the conditions that you had on the screen will one day have implants? Are you thinking, what's your next?
Yes. I didn't mention, by conditions you mean mental health conditions. I didn't mention an OCD study that we collaborated with folks at Stanford and now UPenn. So we built a VR task for them to trigger their compulsions, and they also had a real-world task with eye tracking where they actually introduced these items like rotten fish and so on, and they found a signal there that was sort of differentiating between the two, and then used that to try to reduce compulsions, and it was very successful in one patient, and now they're trying to do more. So these devices are implanted in patients with depression. They're implanted actually in a cohort of individuals in the language areas at San Francisco. They're implanted in adolescents, and more conditions, I imagine, down the line. Oh, Tourette's. So there's a lot of area to move into there. So hopefully others will do it too, because we can't do all of it. But we're sharing the tools with those labs so that they can explore these things. Yeah.
Hi. In your study of the spatial navigation around the UCLA campus, you showed the individual having this much stronger response when outdoors compared to indoors. And this speaks to this larger issue that's going on, outside the fields of neuroscience about the mismatch, the idea that humans have moved indoors and that humans didn't evolve to be indoors. Nature is highly efficacious in reducing many psychiatric symptoms. So I'm just wondering, one thing that goes on in people who are studying this sort of advantage of the outdoors is that some people actually prefer indoors. So there's, in questionnaires and interviews, people say, well, they feel safer indoors, they don't like going outdoors, and so on. Anyways, given that there seems to be this therapeutic benefit of being outdoors, I'm wondering if this tool you have could help to start identifying, because it was such a big signal. I mean, it's one of the strongest effects you had
every
there.
single channel.
If you could use use this tool to help understand the positive benefits of being outdoors, or individual differences in responding to it. Thank you.
That's a really interesting idea, and actually maybe the first I've had to think about this, so I'm thinking as I'm speaking. So that's interesting. I think in order to do that, we need to try to capture a lot of the sort of biometrics that are going on in terms of seeing whether they're ideally pupil size changes or lighting, or we need to control for all these other things and explore this. But I think that would be very interesting, especially in these participants who have implanted in areas that for anxiety disorders or psychiatric disorders. So I think there is a way to look at this. We would just have to look at arousal and other things to try to see whether it's what is really going on and contributing to the signal. One thing we're exploring in that data set, because this is not necessarily a study on arousal or anxiety, but lighting and also eye movements, whether they're making just more eye movements outdoors. There's a lot of, this is the challenge we all face, right? There's a lot of complexity there. But I think there's a way to maybe get into answering that question. I think it's a very interesting one. So I'm of the believer that almost anything is possible after what I've seen in the last 8 years here. So, hey, why not? You just got to think about it really, thoughtfully and capture as much as you can to really rule out as many confounding variables as possible to get to your answer. But it's an interesting idea. Thank you.
Okay. Thank you so much, for all your great questions. Wonderful discussion. And thank you so much again, Nanthia, for starting the session with such an amazing way.